We've all seen those commercials. Are you a man over 40 suffering from low sex drive, fatigue, and depression? Try talking to your doctor about low testosterone. Advertisements like these demonstrate the prevalence for hypogonadism, or low T, amongst men. After all, most men start experiencing symptoms of hypogonadism around the age of 30, and more than 40% of men 45 and older have lower than normal levels of testosterone. In general, there are two types of hypogonadism, both of which affect the HPG axis. The HPG axis refers to the cooperative functions of the hypothalamus in the brain, the pituitary gland, and the gonads. In primary or hypergonadotropic hypogonadism, the gonads fail to respond to the hormones LH and FSH, which are released from the pituitary gland. Normally, these hormones help in reproductive maturation. In secondary hypogonadism, however, otherwise known as hypogonadotropic hypogonadism, the pituitary gland fails to release these hormones. Either type of hypogonadism may be caused by an inherited trait or be acquired through injury or infection, and despite recent medical advances, the basis for many of these inherited cases remains unknown, but luckily a team of researchers may have found the culprit. MicroRNAs are small molecules that can silence genes by inactivating messenger RNAs, which are necessary for translating genetic information into proteins. Because of this, microRNAs are responsible for a range of biological processes, such as cell differentiation and development. Their dysregulation can have serious consequences, and it turns out that the dysregulation of a particular microRNA, microRNA 7A2, leads to secondary hypogonadism. Here's how they did it. Researchers bred mice to lack one of three microRNA 7 precursors, 7A1, 7A2, or 7B. Mice lacking microRNA 7A1 or microRNA 7B were both unaltered in their sexual maturation. Mice lacking microRNA 7A2, however, showed signs of secondary hypogonadism, evidenced by smaller gonads, fewer gametes, and infertility. Next, they wanted to understand the mechanism. In other words, how does knocking out microRNA 7A2 in the pituitary cause these symptoms? To figure this out, researchers compared gene expression of known pituitary genes in mice lacking microRNA 7A2 and control mice not lacking this microRNA. Because microRNAs silence genes, you would expect direct targets of them to be elevated in their absence. Levels of both PTGFRN and GLG1 mRNA were elevated in mice lacking microRNA 7A2, and therefore must be the direct targets of this microRNA. But so what? Well, both of these genes are negative regulators along a pathway that produces FSH and LH. Do you remember those hormones? This means that at high levels, PTGFRN and GLG1 both inhibit puberty. Taken together, these findings demonstrate the importance of microRNAs in human reproductive health. With this information, microRNAs could be utilized for creating new therapeutic techniques for treating individuals with inherited forms of hypogonadism. They should make a commercial for that.